Okay, so let's review for your lab practical. First of all, let me tell you that anything from the pre-labs, the worksheets, or anything we discussed or saw in lab, from all the way that first day safety and introduction, all the way through the tissue lab, can be on the practical. What I'll do is I'll have 50 cards that I set up around the room in a chronological order. You'll be placed at one of those cards that has two questions. A timer will sound every 40 seconds, and you have 40 seconds to answer those two questions. They will be on the same topic. For example, I might have a microscope there and have a blue dot on the ocular and a red dot on the arm, and say identify the blue dot and the red dot, and then have multiple choice answers. So the whole test is multiple choice. You'll find your Scantron. I'll already have your name on it when you come in the room. You'll put all your belongings at the front of the room. Of course, you're going to lay your notebook on my desk so I can check your work. If you don't know what work I'm talking about, check the announcement on Canvas. Once you've found your Scantron, you're going to complete the information on the board, and then you're going to stand there until I give you further instructions. You will begin with wherever I put you. For example, if I put you at 3-4 on your Scantron, you'll start on number 3-4. If I put you on 5758, you'll start on the back of the Scantron on 5758. And then after we have 50 rotations, you'll end up back where you started. My advice is to answer every single one of them the first time around. Circle lightly the numbers you want to go back and look at, because after the timed rotation, then you're going to have about 10 minutes to go back and look at those you have a question about. So you'll want to mark those. My advice is to answer every single one of them the first time around, even if you have to guess at them, because people that leave them blank get confused, and then they look down later, and then they have to get all their numbers mixed up. So you want to answer every single one of them the first time around. So the very first lab, we talked about how lab was handled in safety. So I could ask you any question about safety, and you should be able to tell me the answer. And by the way, if you have not taken your lab safety quiz, please go on Canvas and do that now. So I could ask you, where are the fire extinguishers? What's the first thing you do if there's an accident? Where's the first aid kit, etc. The next lab we did was anatomical terminology. Remember, we started with anatomical positions, so you should be able to describe that. You should be able to label this anatomical man. If I give you the anatomical term like popliteal, you should be able to tell me that's behind the knee. Or if I said back of the elbow, you should be able to tell me olecranon. And again, it's all multiple choice. You need to know directional terms like superior cranial and cephalic is towards the head, inferior and caudal is towards the back. Towards the bottom. Caudal is like a tail of a fish. We usually don't use it in man. You should be able to use these in a sentence too. If I said the eyes are blank to the mouth and gave you superior and inferior as choices, the eyes are superior to the mouth is inferior to the eyes. Anterior means to the front of, ventral means belly side, posterior to the back of, dorsal means bony back. So if I said my spine is blank to my sternum, which is my breastbone, my spinal cord or spinal column is posterior to my sternum, which is anterior. Medial and lateral. If I compare my nose to my ears, my nose is medial, it's more towards the middle, my ears are lateral. But what if I want to compare my nose, my eyes, and my ears? Now my nose is medial, my ears are lateral, and my eyes are intermediate, they're in between. Proximal and distal has to have a point of origin. So if we looked at the arm attaches at the shoulder. So if I compare my elbow and my elbow is proximal, closer to the origin. My wrist is distal, a further distance away. Superficial and deep. Superficial is close to the surface, deep is further away. My skin is superficial to my muscle. My muscle is superficial to my bone, but my bone is deep to my skin and my muscle. You need to know the planes of the body, which we divide body and body parts by. This one divides into top and bottom, so it's transverse. This one divides into front and back, so it's frontal. This one divides into left and right, so it's sagittal. Actually, this one is a mid-sagittal. And one that's at an angle is oblique. The red cavities are ventral cavities. The yellow ones are dorsal cavities. This is the cranial cavity, the vertebral cavity. This is the pleural cavity, pericardial cavity, and both of those are part of the thoracic cavity. Abdominal cavity, pelvic cavity, and the space in the middle of the thoracic cavity is the mediastinal space or mediastinum. Closed cavities are lined with serous membranes. There's always two. The one that lines the space is parietal, the one that covers the organ is visceral. Mucous membranes line open cavities like the oral cavity, the eye, the ear. Cutaneous membrane is the skin. It's the only dry membrane. Synovial membranes are found in joint cavities. They're lacking the epithelia. Sometimes we divide the abdominal pelvic cavity into 4 equal quadrants so that we could describe organs or pain that are occurring in this area with more precision. So we divide it into upper right, upper left, lower right, lower left. Remember, when you're looking at a picture, to swap the right and left. Sometimes we divide the abdominal pelvic into 9 pretty equal areas. This one where the belly button is, of course, is umbilical. Sometimes we call the umbilical our stomach. So above the stomach would be epigastric, below would be hypogastric. Our hip bones are the iliac crest, so this would be the right and left iliac. I slumbar my lumbar when I sleep on my back, so this is the right and left lumbar. And then under the cartilage of the rib cage would be right hypochondriac and left hypochondriac. This is rat, that's liver, stomach, pancreas, small intestine, actually the duodenum, mesentery, the first part of the large intestine, which I say looks like a second stomach, that's the cecum. This is the spleen, this is small intestine, this is large intestine. This is the thymus, this is the heart, the lungs, the diaphragm, the bladder. The one over here with the blue is the female. It has 2 openings right near the tail. This one over here that's on the wood background is a male. You can see the opening to the urogenital opening. You could palpate the penis. This is the scrotal sac with the testes. You'd have to pick it up to see the anus. This is a boy on this side. This is a girl on this side. This is a kidney, testis, seminal vesicle, urinary bladder, penis, kidney, ovary, uterine horn, urinary bladder. Number 1 is the trachea, number 2 is the heart, 3 the lung, 4 the diaphragm. 1 the liver, 2 the stomach, 3 the pancreas, 4 the duodenum or small intestine, 5 small intestine, 6 is large intestine, actually it's the cecum, 7 is the urinary bladder. This is a microscope. You should be able to label all the parts of a microscope, whether it's a picture like you have in your lab manual or it's an actual microscope. You should also know the procedures for finding specimens under the microscope. When do you use a cover slip? When do you use coarse adjustment? When do you use fine adjustment? Your lab worksheet should help you with that. This is a cell much like you labeled for homework in lab. This one gives you all the organelles if you hit the key of the PowerPoint while you're looking at it. And so you should be able to label these organelles. Also on the lab sheet, we got a general function of these organelles. You need to know that as well. This was a squamous epithelial cell. We got it from your cheek. If you notice, there is a nucleus in the center of the cell. There's a plasma membrane around, and anything between the nucleus and the plasma membrane is cytoplasm. The stages of mitosis and the cell cycle. Interphase is not really part of the mitosis, but 2 things happen in interphase. The DNA replicates and so does the centrioles. Second picture would be prophase. The centrioles are at opposite poles. There's asters around them, spindles between them. The nuclear envelope's not quite gone away, but the chromatin is starting to turn into chromosomes. This is metaphase. Anaphase, they're pulling away. 2 daughter cells, telophase. Prophase, metaphase, anaphase, telophase. I will have a microscope set up, and you'll have to tell me what phase is at the pointer under the microscope. We did this lab online. I kind of talked about it a little bit when we were talking about the cell. Some of the factors that affect diffusion are temperature, the amount of the substance, the diffusion medium, like what it's made out of, how thick it is, how deep it is. Also, things that affect is how much substance you have moving and what that substance is. This one we put potassium permanganate and methylene blue. Potassium permanganate is smaller than methylene blue, so it diffused faster. We also had a dialysis bag that we put starch and sugar and salt inside of. We put it in pure water. We let diffusion take place because this membrane is selectively permeable according to size. The salt and the sugar diffused, but the starch did not because it was too big. Starch is a polysaccharide, whereas the sugar is a monosaccharide. We tested the water for starch with Lugol's iodine. We got an orange test, which is negative. Had it been positive, it would have been black. We tested the water for sugar. We got a positive orange test. If it had been negative, it would have been blue. And we tested it for salt. It was positive. It was cloudy. If it had been negative, it would have been clear. We used silver nitrate for salt. We used Benedict's reagent for sugar. We used Lugol's iodine for starch. What kind of tonicity? If I were to put a red blood cell into pure water that had no salt, I would compare my outside to my inside. Outside is less than inside, so it's hypotonic. What's water going to do? Go from the outside in until the cell explodes. So hypotonic, low lysis, the cell is going to lyse. If we put that same cell in 10% salt, that's higher, the outside is higher than the inside, so it's hypertonic. What's going to happen to the water? The water is going to draw out and the cell is going to shrink up or crenate. What if I put the cell 0.9 into 0.9? Compare outside to inside is equal, so it's isotonic. The water is going to be moving in and out at the same rate, so the cell basically doesn't do anything. It stays the same, has the same tone. This is simple squamous, stratified squamous, simple cuboidal, simple columnar, simple squamous around that open circle, simple cuboidal, a bunch of little wagon wheels, each one with simple cuboidal around it. Stratified squamous, actually the top part up here is epidermis and the bottom is dermis because this stratified squamous is skin. This is simple columnar, actually 2 rows of simple columnar. This is hyaline cartilage, adipose, regular fibrous, dense regular fibers. This is blood. This is areolar. It's a loose connective tissue. This is bone or osseous. These are 3 kinds of muscle. The first one is skeletal muscle. It's the only voluntary muscle. It has many peripheral nuclei. It has striations. It's found attached to bones and to skin. The second one is smooth muscle. It's found in hollow organs. It's involuntary. It has a central nucleus. It does not have striations. It looks more like epithelium to me than muscle. The last one is cardiac muscle. You find it in the heart. It's short branched, striated cells. They have a single nucleus and it is involuntary as well. This is skin. A is epidermis, B is dermis, C is hypodermis. It's fat. It's not really a skin layer. Number 1 is the medulla of a hair. Number 2 is an oil gland or sebaceous gland. Number 3 is the erector pili muscle, which makes the hair stand on end to help conserve heat. Number 4 is the cortex of the hair. Number 5 is a sudoriferous or sweat gland. These are some pictures in your lab manuals that I could cut out and have on the lab practical. Study all of your pre-lab questions I assigned, all of the worksheets from safety through tissue, skin, and membranes. Know the safety rules, know how to label and use anatomical terms, know the 11 systems of the body, know the major organs and functions of each. Remember we had that set of matching on the rat sheet. Be able to label rat pictures and specimens, be able to label microscope pictures or the actual microscope, know the proper procedure for handling microscopes. Be able to label the cell mitosis with pictures or models. Remember, all have one phase of mitosis under a microscope. Understand the procedures of the cell physiology lab, what reagents were used, what positive and negative tests look like. Review the tissue types. Be able to identify them from pictures or descriptions. I will not set them up under the microscopes since we did not see them under the microscopes. I will use the pictures from the PowerPoint where you originally saw it or from this PowerPoint. Remember to do your mastering homework. It's due 24 hours before the practical. I'll record it while you're taking the practical. You can do it late, but I'm not going to record it again until the end of the semester and you want a good midterm grade. Please contact me if you have any questions on Canvas.